Hello, good afternoon, children. Today it's my third lecture, and today I will take another new topic, and that is osmosis and diffusion. Dear children, when I began the bio teaching, the first topic was cell, the structural and functional unit. Then I have taken the second one, cell division. And now when we study different type of cell organelles like plasma membrane, nucleus, nucleolus, endoplasmic reticulum, cytoplasm, Golgi body, these cell organelles are covered with a semi-permeable membrane, and that semi-permeable membrane shows a process, that is the movement of the particles from one to region to another region, and that process is called the osmosis and diffusion. In our body also there are the movement of the particles from one region to another region. As you know, the circulation of the blood in the human heart, in the vessels, in the arteries, veins, venues, capillaries, etc. So it is very important in the connection of this cell organelles, the osmosis and diffusion are very important process. Now in home you can see there are many examples of osmosis and diffusion. Now in osmosis and in diffusion, through the activity and through the examples, you can easily understand that what is the osmosis and what is the diffusion. For that, today I am going to explain these two processes with the help of diagrams and with the help of examples. So coming to the topic, very first I will tell you the definition of osmosis and diffusion and differences between osmosis and diffusion, and also with the examples you can easily differentiate, because until unless you are not going to differentiate between osmosis and diffusion, the problem will not be sorted out. So for explaining osmosis and diffusion, the types of osmosis, I will explain the types of osmosis, then I will go through the diagram, and then the topic would be clearly understood. Now, definition of osmosis. Very first, what is osmosis and what is diffusion? Osmosis is a process in which the movement of molecules takes place from dilute to concentrated solution through the semi-permeable membrane. Now, diffusion is also the movement, the process of moving particles from its higher region to their lower region without membrane, that is diffusion. A very important thing, that the osmosis requires plasma membrane, semi-permeable membrane, that is plasma membrane, and diffusion does not require. Diffusion occurs in the solid state, in the liquid, in the gaseous stage, but osmosis takes place only in the liquid state. Now, the osmosis is divided into two types. Very first, you have to understand osmosis. When the water molecules move from dilute to concentrated solution, it means the water molecules move from its higher concentration to its lower concentration. Why they move? Try to understand why the movement takes place. Because due to difference in concentration, at one place The water is 100%. The water is pure water. There is no mixing of any concentrates, solute. And from that 100%, when the water is moving to another side, where the concentrates, where the solutes are there. So water move from its 100% to its 60%, 50%, 70%, whatever you can add there. So this movement from its higher region to its lower region is called osmosis through one semi-permeable membrane. As I'll tell you that this is a pouch, what is going to be happen, I will definitely explain. But before going there, you have to understand what is the hypertonic solution, what is hypotonic solution. Now, the solution which is made up of solute and solvent. It is very important, before beginning the explanation of end osmosis and ex osmosis, I will explain the solution. There are hypertonic solution, there are hypotonic solution, and there are isotonic solution. For explaining the movement, I will explain the very first you understand the definition of these two type of solution, hyper and hypo. Every solution is made up of solute and solvent. Suppose sharbat is a solution, and sharbat there are two things. One is sugar, that is called solute, and in which this sugar is going to be dissolved, that is called solvent. So solution is made up of solute and solvent. Water is solvent, sugar is solute. When there is tonicity, and this tonicity will definitely confirm the movement of molecules from its higher to its lower. After this hypotonic solution, you will able to understand the movement of dilute towards the concentrated solution. I am going to explain the end osmosis and ex osmosis. Very first, look at this end osmosis. In a pouch, I have kept, suppose, A and B. Inside pouch, I have kept the hypertonic solution. Okay? And in the B, the tonicity is hypotonic. You stay here, that inside hypertonicity and outside hypotonicity. Hypertonicity means concentrated. It is thicker solution. It will not move. The hypertonic, again come to the topic. Hypertonic, A is hypertonic, and this A is having more solute, and on the other hand the B will have less solute. It means it is thin. The hypotonic is thinner and hyper is thicker. Okay? Got my point? You stay here. That why this movement will take place? Because it's a tendency of molecules that they move from their higher region to their lower region till its equilibrium. It will not continue for a longer duration. When the equilibrium means inside and outside solution will be equal, will be isotonic. When it will be reaching to the isotonicity, this will be stopped. So now see, suppose in hypertonic solution there are 60%, 80% solute, and in the hypotonic solution the solvent is moved. So the inflow of the water is taking place. And with the example of dry resin, dry kismis, you can easily understand after 1 hour what you observe. The same process you are going to observe. The dry kismis, dry resin will swell. Why that is? There is end osmosis, because dry resin inside tonicity is hypertonic. It contains less water, and that is why it will absorb. The movement of water will be taking place inside, and after this what will happen? What will be the volume of this pouch? It will swell. It will swell the volume, because the water is moving from B solution to A solution. The number of water is more in the B, the water particles are less in the A. Automatically it will swell, it will absorb, the volume of A pouch will be increasing. Got my point? A very good example of this, that when you put dry resin, dry kismis in any particular glass where the water is there, after 1 hour you will observe that the pouch is, that dry resin is going to swell. And what will be the result after this end osmosis? Entry of water molecules in this A is indicating that it will swell, it will rise in shape size. The volume will increase in the pouch. Right? So here once again I am going for the recapitulation that what happened. The tonicity of A pouch is hypertonic, and the B, where the pure water is there, that is hypotonic solution. You can see hypotonic solution. So water will move from B to A, it will swell, and the swelling will take place. On the opposite to this end osmosis, now I am going to see towards the X osmosis. Same diagram. Take a beaker, fill it with the water, again I am going to label this A and B. But this time what is the change? Look at this. Here A will be, what will be? A will be hypotonic. And B tonicity would be hyper. I am just going to do the opposite work of case 1 and case 2. Both are opposite to each other. Here the end osmosis entry is taking place, and here the exit of water will take place. Why the exit of water will take place? Because A tonicity will be the hypotonic, means the water will be 100%. Water will be 100%. And here the water will be in the B. What is the nature of B? Nature of B is here hypertonic. You need not to do any extra thing, any extra exercise for the keeping these two things in your mind for your memory box. You can easily understand these things. The percentage of water is more at A, it is lesser. It's a quite natural thing. The water will move from inside to outside, and that is why it is called X osmosis. Exit. Here entry and here exit. Now you will easily understand that the exit of water is resulting into the shrinking process. What will happen after this exit? That this will shrink. Here in end osmosis the swelling takes place, and here it is shrinking. Crenation, when we keep a drop of blood and surrounded by the salt solution, it exits. It is the process in when we preserve our pickles, we keep the salts in that pickles. The X osmosis takes place, the exit of water takes place, the moisture is removed, and that is why pickles are saved for longer duration. Now come to this. Now, shrinking takes place. What will be the result? Through the arrow, you are looking that A tonicity is the hypotonic, having more water. Here in the B, in the beaker, the percentage of water is less. That is why water will move from inside to outside, and it is going to be proved. X osmosis, shrinking takes place. Now, in this lecture, you got the differences between end osmosis and osmosis. Now, when we are moving for the diffusion process, when we are burning an agarbatti at the corner, the smoke flow from that corner to another region. Due to difference in the concentration, when at that particular place we are burning the agarbatti, the smoke will flow from that side to another side, because the smoke, the percentage of smoke is more at one side and it is lesser at another side. That is why it will move from its higher concentration to its lower concentration till its equilibrium. And in the diffusion, when we are putting diffusion, there is no need of plasma membrane like this. In this osmosis, plasma membrane is very much required, and in the diffusion, there is no need of plasma membrane. Right? So, in diffusion process, when we are putting a chalk powder, a solid substances, a ink color in water, that diffuses, that is spread. So there is no need of any membrane in the diffusion. Only this is one main difference between diffusion and osmosis. The movement of particles occurring from higher region to lower region in both cases. But in the diffusion process, there is no need of a requirement of the plasma membrane or semi-permeable membrane. So now I am sure that you got it very clearly. If any doubt would be there in your mind, you are free to ask any questions, right? So after these two explanation, end osmosis and X osmosis, you can go through the NCERT textbook. You have to draw these end osmosis and X osmosis diagram. It is occurring through the semi-permeable membrane, and that is very important for your annual exam, for your half yearly also, for the complete preparation of the end osmosis, X osmosis. You have to prepare at least 4 or 5 questions I am going to give you for the assignment today, for your homework today. For revision, I'm going to give you 2-3 questions now. Differentiate between osmosis diffusion. Question number 2. Differentiate between osmosis and X osmosis. And last question, dear student, now you will prepare after the lecture. You have to see these 3 questions. On the basis of these 3 questions, you can complete your this osmosis and diffusion topic. First is differentiate between osmosis and diffusion process. Second one is differentiate between end osmosis and X osmosis process. And third one is what is hypertonic and hypotonic solution. For today's now this matter is sufficient for today. I will be meeting next day with next lecture. Thank you very much.